we discovered it. There was a problem in your protein, the last portion of it. It was dysfunctional here. It was different, wrong sequence, and there was early publication sequence right there. And to find out exactly what the problem was there, we looked at chromosome number 13. And here's the section here, our stages, and we find that the problem in Mary's gene is there. And we lost a nucleotide here. There's supposed to be a G here. And how is it that that single loss of nucleotide caused all the problems that we see in that cell? Why is it that that whole protein, even though we only lost one nucleotide, why did the whole protein get changed? Because it causes frame shift. Exactly. Frame shift begins where the word breaks occur, and if you change one of these things, you get a type of division called point deletion. You tend to get these frame shifts that will cause massive changes in the way things are understood by the cell. And I gave you an example of that last time. Now, this is just one type of mutation that I want you to know. I'm going to give you a list of some of the other mutations that we have. These are not entirely exhaustive by any stretch. There's a whole bunch of types of mutations we have. But this one is just an example we call point deletion. Why is it called point? Yeah, one single nucleotide. So one single nucleotide is lost as what's called point. In addition to that, we also have something called point insertion. What's going on there? You guys?
An additional one?
Right. You're adding one single additional new nucleotide. Only one, that's why it's point. We also can have point substitution. What's going on there? Right, so I remove one, put in another one. So A gets changed to C, C gets changed to D, or something like that. And so that would be these would be point substitutions. But again, it's only a single one, so that's the point. These can also, that essentially can happen to groups, to large groups of nucleotides, in which case we call it a block. So the first two would cause a frame shift. Good question. I was going to say that later, but I should say because that's exactly what I want to get into. This we know can cause a frame shift. And this, if you think about it, you can figure it out. Yeah, you bet. So these two both can cause frame shift. What about this? Now, point substitution typically does not. In fact, it can't. So point substitutions typically have a much smaller effect on the phenotype than either point deletion or point insertion. Good, because these guys are frame shift generators, these guys aren't. Okay, now there's also this loss deletion. That's what I'll call it. So what do you think that is?
Removal of a word.
Right, a whole bunch. You get a whole host of nucleotides get removed. And this block can be any size. It can be thousands of nucleotides. Right? Can that cause a frame shift? Yes. It can also not cause a frame shift. It depends. If it's a multiple of 3, if the number of nucleotides that are removed is a multiple of 3, it doesn't necessarily have to cause a frame shift. We'll see why that's true in just a moment. And then we also then have block insertions. And where you get that? Right, so now a bunch of nucleotides are inserted into the DNA. Okay? And again, this may or may not cause frame shift. Depends. If this is a multiple of 3, it doesn't necessarily have to be a frame shift. Okay, so then there's this type. And I'll show you an example. Tell me what's going on. So here I've got this sequence. I've got 5' here, A, C, A, G, G, A, G, 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 C. Okay, now, you know the rest. What goes here? A prime, 3', good. And then what goes here? T, then G, then P, C, C, D, A, C, D. What goes there? 5'. Good. What goes here? 3'. You got it. Okay. Now, that is mutated to this. We'll just look at well with open. 5' is mutated to A, G, C, C, A, G, G, A, C, C. There's 3'. Okay, I'm only going to show the sense strand here. Now look at that and tell me what happened. I'll make it a little bit easier. Just look at the sense strand. Compare the sense strand. Simple pattern. Yes, exactly. Notice, A didn't change. But G, T, T, A, G, G, A, C, and then last C didn't change. Okay, you see that? It's in reverse. Okay, so we'll call that a reversal. That happens as well. Okay, so that kind of thing. Well, that comes straight here.
No.
Typically not. Right? Good. Okay, so that then is a small one. Not all of them. Those are the sort of the kind of the more common types of mutations you're going to find. Now, this is a single deletion, point deletion here to solve all these problems. Now we want to try to understand why. And what is it about smoking that causes that? Well, smoking is associated with all kinds of changes in the DNA. Sometimes they're mutations, sometimes not. What exactly is a mutation? What is the definition? Here's some types. These are example mutations. So what is the definition of a mutation? Change in DNA, good. Abnormal change in DNA. Okay, so what's the difference? What's the difference between normal changes and abnormal?
That's supposed to happen.
Okay, it's not supposed to happen? Okay, good. Good. That's actually part of it. So, because you can change DNA in a particular way that is supposed to happen, the immune cells do this, which are part of the You can also change DNA in a way that's not mutations. And those are changes in the promoters that we see where you put particular types of groups, either acyl groups or methyl groups, onto the promoters. And that will turn the promoter on and off. That can be normal, and so bad or not. The abnormal can cause cancer. In fact, it's associated with this type of cancer. That is mutation. The reason it's not mutation is you can take those groups off, and everything goes back to normal. So the difference between a change in DNA and a mutation is exactly this. The mutation is a permanent change in the DNA sequence. So permanence has to be part of this. You can change DNA in a non-permanent way, in a reversible way. And that is considered a mutation. Still a change in DNA. It can still cause cancer and the other things, but it's not mutation. Mutation has to be permanent changes in the DNA sequence. Smoking does that. Smoking causes changes in the DNA sequence by damaging cells, by causing cells to have to reproduce a bunch of times. When they reproduce, they cancel the signal on top of the DNA. There are things in the actual smoke that will cause mutations directly. There's things that cause these non-mutation changes. So those are all sorts of things that are listed on the board of the slide. I'm not really going to really get into it. If you want to see that, there's a bonus one to exam. Well, the reason I'm not going to get into it is because I need to get into the next section, which is going to occupy us for most of the rest of the day. And that's this concept. To understand precisely why it is that protein got destroyed, and the way that it got destroyed, you have to be able to read the DNA. So if you look at that protein sequence in Mary's cell, and you want to see exactly why is it that it changed, you cannot see that until you can read DNA. That's what you're going to learn this week. At the end of this week, you're going to be able to read DNA. Read the protein. In fact, by the end of today, I'm going to give you an example. I want to see if you can see the pattern. This is an actual sequence of nucleotides that's found in the human hemoglobin gene. We talked about this before. Remember ATG? That was a signal sequence. What does that mean?
Beginning.
It's the beginning of
The sequence of the meat of the DNA. The information.
the information. Good. Now, what information? Is it the beginning of the gene? In other words, is it the beginning of the promoter?
No.
No. So it's not the beginning of the gene. That's not a start gene sequence. That's the start of what sequence? Precisely. Coding region. Right, that's the beginning of the coding region. Okay, so this is the start of the coding region. Right there. And what that sequence means is the following. Make a protein that starts off with the following amino acids. Methionine, valine, histidine, leucine, threonine, all of amino acids. And that sequence up there, that DNA sequence, helps to make that sequence of amino acids. Okay, here's another section. Another part of the exon, you've got this sequence. And that means to make this. Valine, valine, alanine, glycine, valine. Do you see the fact? What sequence of nucleotides means the amino acid valine? GTG. Good, excellent. GTG. You got it. There it is. GTG, valine. Another GTG, valine. GTG at the end, valine. So you see that? That's the point. There are, for each one of these sequences of three nucleotides, that translates directly into some particular amino acid. Okay, so that's the exact one-for-one mapping. So it's not why exactly why multiple three block deletions, block insertions don't cause frame shifts. Because every single word, unlike English or any other language on the planet, right? Words are not a fixed, in this case, are a fixed number of letters. So for in this case, every single word is 3 letters. And every set of 3 letters in the reading board, called reading frame, code for a particular amino acid. We call that a codon. So a word, the concept of word here is equal to a codon. So each one of these things is a codon. So each codon is exactly 3 letters long. Each word is 3 letters long. Okay? So that makes it easy. So you don't have to worry about word breaks. The cell knows that everything's 3 nucleotides in length. So now you just have to learn how to read it. Okay, how do you do that? Well, the first thing you got to do is this. Let's go back to the actual mechanism. Let's start getting into the mechanism today. How is it that you've got to get the information? We're thinking conceptual. Remember, you want to make the Kraft puff. Kraft puff is a protein. You're what? Kraft puff is a protein. Ribosome. You're a ribosome. Ribosome can make that protein. The information on how to make the protein, how to make the Kraft puff, was held where? In the library, the nucleus. So you're outside the nucleus, the information you need to fill the nucleus. How did you solve that problem? Yeah, messenger comes and brings you the information. Messenger is like RNA. So you've got to have this event occur first before you can make the protein.